This conference will now be recorded.
Okay, excellent. To start this webinar, which is the first of what we hope will be a series that will be repeated in conjunction in between the Italian Cristallographic Association section on biological macromolecules and the Italian Biochemical Society proteins group, protein structure and function group. Of course, the two interest groups have common interests and common aims, so it makes a lot of sense to share experience, to promote interactions, and hopefully get some synergy in between the two interest groups. The format we have decided so far is to have just two presentations, to have a sort of light webinar, which can be followed upon a cup of tea or something in the afternoon. And the presentations in general will be given by scientists belonging to the two groups, one junior scientist and one senior scientist. We will see how it works, and we will hopefully continue this in the coming year, coming months, coming years. So to move into the real presentation, that will be the first one will be given by Dr. Christina Ginovich. It's my pleasure to introduce Christina, who is a friend, has been a friend for many years. I do not count them because that will be bad for me. Christina is currently head of the Department of Structural and Computational Biology at the Max Perutz Labs at the University of Vienna. Christina comes from Slovenia, from Ljubljana. She passed as a, I would say, a typical Central European person. She spent times in different Central European countries, such as Italy, in Pavia and in Trieste, Germany, in Heidelberg, and then in Vienna. Of course, not counting as well Slovenia. Christina has an impressive CV, rich of citations, publications, invited talks, grants, by the way, and the focus of her research is, of course, structural biology based on X-ray crystallography, with specific topics on actin-based cytoskeleton, and specifically the muscle Z disc, but also from the times she was in Italy, there's some substantial interest for metal enzymes involved in oxidative and oxidative damage and protection from oxidative damage. Also, she has some interest in X-ray induced radiation damage that for X-ray crystallography is always an issue. So I should limit myself to this, but I would say her career is really impressive, and through very important groups throughout Europe. So I wish to thank Christina particularly for being with us today, and I invite her to start her presentation.
Thank you very much for this very nice and kind introduction, Martino. I'm, of course, very flattered to have the occasion to talk in front of this audience. It's also very nice and touching to see many people who with whom I worked and with whom I met during my times in Italy. And it is, of course, very flattering, but also a little bit making me a little bit nervous talking in front of Martino, who actually initiated me to structural biology. This is where I learned macromolecular crystallography and was initiated to structural biology. So thank you again for this occasion. And I would like to start with I would say, textbook knowledge or introduction with the textbook knowledge to my talk, and that is to repolish and to put us all on the same standing point about the striated muscles. So if we look at the striated muscle as an organ, you will see that it is composed of a bundle of muscle fibers, which are in turn composed of each muscle fiber is actually a cell, which is in turn composed of a number of myofibrils. And the basic building block of myofibrils is the sarcomere, which, as seen here in a microscope, is striated. And these striations actually come from different protein densities throughout the sarcomere. This is a schematic representation of the sarcomeric filaments. You can see here myosin. You can see here actin filament. The sliding of myosin along actin leads to the contraction. You can also see here titin, the longest protein on the planet Earth that determines the length of the sarcomere, and is the blueprint of its assembly. We are not actually interested in the contractile machinery, which is myosin sliding along actin filaments. What we are interested in is this area here, which is the Z discs. This is where the actin filaments from adjacent sarcomeres, and these are antiparallel actin filaments, are cross-linked together and fixed in a very organized and structured assembly. Also, titin is fixed in this area here. So this is now a more zoomed-in representation of the Z disc, where you can see antiparallel actin filaments cross-linked with this major protein of the Z disc called alpha-actinin. So this cross-linking of actin filaments is important not only for the contraction itself, because if everything were moving, of course you wouldn't get anywhere, but also the organization and precise organization of the Z disc is also important for the correct transmission of the force generated in one sarcomere to the next sarcomere and throughout the myofibril in order to actually obtain a macroscopic effect, a contraction at macroscopic level. So if we look at the Z discs in microscope again, but this time in transversal section, so transversal section here, you will see that this is a highly ordered system. You can see, you can recognize tetragonal symmetry where one actin filament is surrounded by 4 actin filaments which are of the opposite orientation. And this proteinaceous material between actin filaments is actually alpha-actinin. If you do this under the contraction, you will also see organized system with a bit different organization, also called weaved basket. I've always, when thinking about this, find it really amazing that in your relaxed muscles now, you have this paracrystalline organization of actin filaments in your Z discs. So this is the number of crosslinks, the number of alpha-actinins which crosslink actin filaments can go from 2 up to 4 or even more, and this depends on the type of the muscle. Slow muscles have more of these crosslinks, fast muscles have less, and how many will actually there be, it depends actually on titin, because alpha-actinin is targeted, as you will see, to the Z disc via specific interactions to these titin Z repeats, and how many of these titin Z repeats we have determines then how many crosslinks we will have here. So our long-term aim is to understand the molecular architecture and assembly of the Z discs of striated muscle. It's known that the sarcomere biogenesis is a hierarchical process which starts from the Z bodies, which are assemblies of 5 to 6 proteins. One of them is alpha-actinin. Of course, we also have actin here and several other all Z disc proteins which are present there, as well as in the mature Z discs. So we are asking ourselves, what's the hierarchy and cooperativity of this assembly? How are these diverse structural properties in sarcomeric Z discs assembled in a mechanostable and at the same time plastic complex that is able to transmit force along myofibrils? And finally, we're also looking into selected disease mutations that disrupt finely tuned assembly. So how was the molecular basis of disease in essence. There are already quite some structures of Z-disc proteins and some complexes, but there's still a long way to go, I think. So the approach we are taking is integrative structural biology approach. As Martino said, my basic and fundamental training is in macromolecular crystallography, but we are now embarked also on other approaches from small angular X-ray scattering, NMR. We are going towards microscopy as well, combined with other biochemical and biophysical complementary approaches. So alpha-actinin is the major protein of the Z-disc. And if you take alpha-actinin and simply expose it to actin filaments or expose actin filaments to alpha-actinin, you will see that actin filaments are going to be cross-linked with alpha-actinins all along and throughout the entire length of actin filament. On the other hand, we don't see this in the muscle. We see that in the muscle, these cross-links only happen in the Z-disc. Why is this? We believe that this is because alpha-actinin is targeted to the Z-disc through specific interactions that are interactions to these specific regions of titin called Z repeats. The mechanism that was proposed goes as follows. Alpha-actinin, which is an anti-parallel dimer, which you can schematically see here, composed of actin binding domains, spectrin-like repeats, and calmodulin-like domain, which does not bind calcium. In this anti-parallel organization, calmodulin-like domain binds to this juxtaposed neck region. And in this conformation, it is not able to bind actin filaments. This isn't able to bind titin, but it is activated by, or regulated, if you want, by PIP2, which binds to actin binding domain with this polar tail, opens this interaction here, therefore leads to opening the calmodulin-like domain, which is now ready to bind to titin, which you can see here. If we now look at the sequence of Z repeats and compare it to the sequence of the alpha-actinin neck region, which is this region here, you will see that there is a certain sequence similarity, and that's the reason why this mechanism of regulation was coined phospholipid regulated intramolecular pseudoligand mechanism. I think it says that this was coined by a German speaker. This is a concatenation of words. But in order to really understand this mechanism, we set for a structural determination of alpha-actinin. This is because we are here amongst, this is a crystallographic meeting. I will just spend 2 slides on structural determination of this human alpha-actinin structure. First of all, we did need to do some, I would say, massaging of the construct and of the sample by truncating the N-terminal region, which was disordered, by optimizing the buffer to enhance protein stability, by then further optimizing the sample by protein surface engineering, on the one hand, protein reductive methylation, and on the second hand, surface entropy reducing mutations. One of the 3 triple mutations that we designed actually worked, in the sense that we had about 1% of crystals that actually diffracted. We collected 15 datasets from 2 different crystals, which led, after merging everything together, to 3.5 angstrom resolution and a multiplicity of 100, which actually led to this resolution. This is the structure of alpha-actinin in the closed conformation, where you can recognize this actin binding domain, calmodulin binding domain, which is exactly binding to this neck region as the model was suggesting. And of course, this is a dimer, an anti-parallel dimer. You can see the same situation here. I always like to compare this central region to the fusilli structure. Now, in order to understand better how the protein now works, we designed 2 types of mutations. One is the mutation where we abolished the binding site of PIP2. That would therefore be non-activatable PIP2 mutant. It couldn't bind. PIP2 would therefore be always closed and not being able to bind to actin, to titin, and this is what we actually see here. We much abolished the binding to titin compared to the wild type. This is the mutant. Then we also made another, which would be constitutively active mutant, where we made mutations in this neck region here in order for this carboxyl ligand domain EF-hence3-4 to prevent binding, so to be open. This mutant I will call the NIC mutant. This is how the sequence read after we mutated it. And we hypothesized, we hoped that we made this kind of an open structure, which would be, you would not need PIP2 in order to be activated and to bind to actin filaments. So here I'm now summarizing in one slide a lot of work of quite some time. And this is, first we showed with molecular X-ray scattering and double electron resonance experiments that this NIC mutant is indeed open. So the calmodulin ligand domain is not bound to this neck region anymore. We showed in binding assays that this mutant binds better to the titin compared to the wild type. We also showed that while the wild type needs to be activated with PIP2 in order to bind to with higher affinity, the PIP mutant, which we inactivated, could not be activated by PIP2 itself. We also showed that in actin co-sedimentation assays that this NIC mutant still binds to actin filaments, so it co-sediments with them, but it also brings with itself titin, Z repeat. This is a very small fragment, and that's the reason why it has also quite weak here, but suggesting that this open conformation actually binds to actin and to titin at the same time, which is what should be happening in the Z disk. Finally, we went into cells with cell biophysics in collaboration with Matthias Gautel. We actually looked at FRAP and with FRAP and dynamics, and we saw that this NIC mutant, which is constitutively open, is very tightly bound, much more than the wild type, is fossilized basically to the Z disk, because of course it cannot close anymore. It is therefore tightly associated with this. And if we transfect the neonatal red cardiomyocytes with this mutant here, we see that we lose the striation on order of compared to the wild type, meaning that this regulation is really important for the assembly. So the last thing we looked at is then actin binding to alpha-actinic to actin filaments. So we took actin filaments decorated with actin binding domain. We took our structure, we superimposed it. This is superposition in PowerPoint here, but we did then this on both sides, so superimposing actin on this side and on this side here. And what we see is actually we got is this structure, this assembly here, which is of course wrong, because actin filaments here are perpendicular and not parallel as they should be, because there's no perpendicular actin filament in the Z disk. So what we think actually this is due to is because in the closed structure of alpha-actinin, when calmodulin domain is bound here, we see this twist throughout this rod part, which is of about 90 degrees. And this leads to orientation of these actin binding sites by 90 degrees. What happens in the Z disk is therefore, we believe, the following. Just have to see whether this will actually start. What we see is, what we believe is going on is that we have this closed structure, then PIP2 will bind, open it. Now, because when this is unbound, this neck region unfolds. We have the data for this, which I didn't show you. This allows now these actin bind domains to reorient as they need in order to be able to bind anti-parallel actin filaments. This is a model that we generated knowing also factoring in information about from electron microscopy. And what we actually in this model also see is that the distance that we obtain between actin filaments is about 200 angstrom, 20 nanometers, which is corresponding to the distances that we actually see in the Z disks, in mature Z disks. So we actually need dynamics and structural plasticity for alpha-actinin to be a structural protein in the Z disk, which maybe is a little bit counterintuitive, but these conformational changes are needed for alpha-actinin to actually bind to actin filaments and to titin. So next thing we then looked at is what is the mechanics of alpha-actinin-titin interaction. See is that the affinities of titin Z repeats to EF hands are in the order of micromolar, while the forces in the muscle under contraction are about 200 piconewton. Therefore, the question arises, how is the task of firmly anchoring actin titin within the Z disk under applied mechanical forces achieved? You can see here that how we imagine the Z disk with these Z repeats to which alpha-actinin bind highlighted here. So each of these interactions is in low micromolar affinity. So in order to address this, we joined forces with Matthias Riff and did optical tweezers experiments where we used EF hands 3-4 in chimera with the Z repeat 7 of alpha-actinin, and we start pulling this construct in this direction. Here we have the force which propagates throughout the alpha-actinin and EF hands 3-4 and Z repeat. So what we see is these unfolding events here at the forces of about 4 piconewtons, and if we apply a constant force, we see again of about 3.7 piconewtons. We see these unbinding events of EF of titin or unfolding of the entire chimeric construct. Using competition assays with the free peptide, we could also determine the KD, which is about 4 micromolar, and about and also K-ons and K-offs. We did then this for all other repeats, which are 7, and this is summarized here, where we see that we have some strongly binding repeats. This was also known before from other studies. And we also have some weakly binding repeats. And also we see that the neck region of alpha-actinin also binds weakly, which makes sense because it has to bind weaker. Otherwise, it would be competing too much with these titin Z repeats here. By the time we did all this, we also understood that we actually had a problem with the geometry of the construct that we used, because we were pulling along the titin and alpha-actinin, while the force in the Z disk is actually along actin filaments and along titin and not along alpha-actinin. So we redesigned this now in the way that we were pulling along actin, titin, Z repeat alone. And what we saw here is that this construction actually needs more force, 12 piconewton for unfolding, and again with the competition, we could determine also the KD of about 8 micromolar. So what we believe is going on is the following. On the first, it's clear that the system is custom tuned to endure force along titin direction and not perpendicular to it, because you can see that this, if we pull a long titin direction, this system endures 13 piconewton compared to 3.5 if we are pulling in this direction here. Secondly, from the kinetic data, we see that at each and every time point, we have at least one titin molecule bound to one of the titin high affinity titin binding sites, since the residing times on titin are a few seconds. Therefore, we believe that we have an avidity effect at work, because each of these interactions is indeed weak, but the interaction-free energies sum up due to the increased valency, leading to basically multiplication of binding affinities. So we have these dynamic bonds that act together to lead to a long-term and stable anchor. So the last part of my talk I will dedicate to fats, also called calcartin or myosin, on which I actually started to work back in my times in Trieste in collaboration with Georgine Faulkner, who actually discovered this protein. It binds to the Z disc, it binds to many Z disc proteins, as you can see here, and one of them is alpha-actinin, as also highlighted in the alpha-actinin interactome. It is also present in the early stages of myofibrillogenesis. It is part of these Z bodies that I mentioned at the beginning of the talk. So bioinformatics analysis shows that fats is intrinsically disordered protein. So when we started to work on it, it was these intrinsically disordered proteins were actually not so known at that time. And so we spent a huge amount of time trying to understand why it is so obnoxiously behaving. And it took a lot of time to actually come to constructs that could be used in biochemical and structural studies. And we actually resorted finally to erase-a-base approach, which does not depend on the bioinformatics analysis. And I'm happy to talk about this later on in the discussion. So this brought us to two constructs that allowed us to move on, and this is one N-terminal and one C-terminal constructs presented here. So molecular X-ray scattering showed that these are intrinsically disordered, but you can see this from the Kratky plot as well as from the analysis of the scattering data, where you can see that both N-terminal as well as the C-terminal construct we can only model as ensemble of unstructured structures, where they are slightly more compact in both cases than the random pool. We then looked at how does fats bind to alpha-actinin. Alpha-actinin is a dimer, so one expects two fats is binding to alpha-actinin antiparallel dimer, and this is in fact what we see. We see two binding events, one nanomolar and one low micromolar affinity. If we use half dimer, we see one binding event, which makes sense. And if we use mutants, excuse me, of fats which were designed to abolish binding, we did see much reduced affinity. Analysis of energetics shows that this is enthalpically driven reaction, to which I will come on later. We then tried to map the binding site with NMR. You can see this is the NMR spectrum of the free delta 91 and N-terminal fats, and this is the delta 91 in complex with alpha-actinin. We see reduction of number of peaks that is due to part of alpha-actinin binding to fats, and therefore these residues tumbling bound to alpha-actinin is reduced, and therefore their signal in the NMR spectrum reduced. Mapping of the binding site was attempted by NMR. What we actually unfortunately could not assign is this region here, which was exactly the region where we thought that the alpha-actinin actually binds. So although we do not know which residue is which here, we do see that drastic decrease in the signal. You can see here I bound divided by intensity of the free, suggesting that this region is involved in binding to alpha-actinin. We then asked, does fat get structured upon binding to alpha-actinin? We use CD here. This is the CD of the rod alone. This is the CD of fats alone, which is a classical signal for intrinsically disordered region. And this is the CD of the complex measured. And in red is the theoretical complex CD of theoretical complex if we sum the CDs of the two free proteins, suggesting that alpha-actinin binding to fats does not promote secondary structure formation of fats. So at this point, I actually managed to convince somebody in the lab to crystallize the complex. We actually got crystals of 3 different complexes, half dimer with delta 91, and then also the raw domain alone with delta 91 and also shorter construct of fats. These are the structures. We started with this one, which is a 2.7 angstrom resolution. This allows us to actually, with combination of also selenomethionine labeling and chemical cross-linking mass spec, to actually assign the sequence to these fats parts that we saw bound to alpha-actinin. We actually selenomethionine labeled fats and then use anomalous difference Fourier to localize the methionine, and therefore this allowed us to be more convinced about the assigned sequence. You can see here that fats binds with 2 stretches that we see electron density of. These are called also linear motifs. All the rest is unvisible in electron density. And these linear motifs can be also termed molecular recognition motifs, as Silvia Longhi actually calls them, or termed them. These are motifs of which dynamics is reduced upon binding to a binding partner. Now, we looked then in what is the contribution of these inner motifs to binding, and you can see here from the isotherms that LM2 is the major contributor, while the LM1 alone does not bind strongly. Nevertheless, we believe that it is a transient binder, and this we also can see in the molecular dynamics, where we saw that, just have to start this, where we saw that if we start from a position conformation where the LM1 is not bound, it actually, in the course of the simulation, lands on the rod of alpha-actinin, suggesting these transient binding interactions. So in order to see where the rest of alpha-actinin is then bound, where is the rest of fats, we use molecular X-ray scattering, and you can see here that we can again model this as an ensemble, which is called in this say business of intrinsically disordered proteins a fuzzy complex, because the only part that is the part that is bound is fixed. The rest continues to be an intrinsically disordered ensemble. So combining this all information together, we actually then made a integrative model of full-length alpha-actinin bound to fats. You can see here binding these LM motifs. These are the structural data from crystallography. And now we put on the full-length alpha-actinin ensemble of fats as bound, as found in molecular X-ray scattering. You can also see here from this orientation that fats actually binds on one side of alpha-actinin rod on this concave site, and it is bound in irradiating in different directions from that site. This also seen that this is, you can see here, this is a polar complex, because alpha-actinin is decorated only on one side of its rod domain. So if we now take the structure of Z-disc, which were recently at low resolution done with cryo-electron tomography, and use our molecular model of alpha-actinin on and fats, we generate this model here. And here I would like to bring you now into the, let's say, virtual 3D model of Z-disc, where you can see these actin filaments. It goes in one direction, surrounded by 4 actin filaments going in opposite directions. You can see these white filaments here, and they are cross-linked by alpha-actinin. Alpha-actinin is bound to actin filaments with this actin binding domain. You can see here one, and the other one will be here. And you can see that these cross links are several. So each central filament is connected by several alpha-actinins to the other actin filament going in the opposite direction. So here I'm showing also fats, which is, as you can see here, oriented in one particular site of alpha-actinin rod. So what I would like to, before I finish with the alpha-actinin fats, I would just like to say that this polar orientation actually we believe is important. Polar assembly is important because it will further orient other proteins that bind to fats in the Z disk, in the final mature Z disk. So the last few minutes I will spend on a topic which emerged recently in the lab, and that is everybody is now talking about liquid condensate. So we looked at the sequence of fats and we found that it actually has a high propensity for formation of liquid condensates. We tried this and it does indeed. It forms liquid droplets which also merge together as they should. We then checked what is actually the responsible part of alpha-actinin of fats for liquid droplet formation. So we can see that the N-terminal part binds, makes liquid droplets. The full length does, although we had to use the fusion because it was otherwise difficult to work with it. The N-terminal does not, suggesting that this liquid-liquid phase separation function resides in the C-terminal part of the protein. We checked the properties of the droplets with FRAP. This is a recovery after photo bleaching, shows that we are now determining the diffusion coefficient of fats within the droplet, and you can see that it is about 0.96, which is well in the range of the phase separated proteins. Finally, we looked into what kind of forces could be at the basis of this liquid droplet separation, and we saw that we can abolish liquid droplet formation with arginine, which is a classical quencher of the cation pi interaction, suggesting that this is the basis of the liquid droplet formation in terms of interactions. We then asked, does alpha-actinin go in these liquid droplets? We have now red labeled alpha-actinin, green labeled fats, and you can see that yes, it does. This is the merge. If we now photo bleach this droplet here, we can see that alpha-actinin, which is red, goes back into this fats droplet, suggesting that the protein mesh in the fats is permeable for alpha-actinin. So this now led us to this, I would say, question: can we actually regulate the liquid droplet formation with the amount of alpha-actinin that we add? And indeed we can, and that is that we can actually dissolve liquid droplets with increasing amount of alpha-actinin. And that is important because liquid droplets are of course not present in the Z disks. We believe that it's the amounts of these binding partners that actually define the phase diagram and the existence or not of liquid droplets. So this brings us to this, I would say, tantalizing hypothesis or question, and that is, does sarcomere biogenesis actually start from phase separation from these Z bodies? Are the Z bodies actually macromolecular condensates? As it was already shown many years ago, back in 90s, these are punctate aggregates that are lined here, and I think this thread is actually actin on which it declines. Now the question we are asking ourselves, is this maybe a liquid condensate, where FAS would serve as some sort of an aggregator or accumulator of the basic proteins that are needed to start the biogenesis, and then with increasing concentrations of these components would actually, droplets would dissolve and lead to the mature sarcomeres. Of course, we opened the Pandora box here, and there's a lot to do to actually validate this hypothesis. So I would like to finish with a dream. I would say I have a dream, and that is to convert this blobological representation of Z-disk into a more structural presentation and representation understanding of the Z-disk. And there is still a long way to go, and I can only go there and along this way in good company of my colleagues and several important collaborators, and of course funding. And with this, I would like to thank you and be happy to answer your questions.
Thank you, Christina. It was a fantastic presentation. I would say a real integrative approach to the structure of Z-disk and sarcomere in general. So even the last few slides showed a sort of future view of what we are looking forward to. So I think we can take a couple of questions or even more perhaps from the audience. I'm not familiar with this platform, so I don't see a way to raise hands. So probably you should try and enter your question by shouting or activating your camera and raising your hand physically in front of the camera. Okay, I'm trying to view everyone. Let's see. Okay, I'm still fighting with a view. Okay, questions from the audience? Let's see here. Okay. Pietro, you have a question?
Yes.
Yes, go ahead.
So first of all, it's very impressive work and a real feat of biochemistry and structural biology. I'm interested whether cryo-electron microscopy offers any route to some of the questions that you have, and whether you've started this kind of work already.
Yes. So yeah, of course, cryo-electron microscopy is offers many routes. On the one hand, the structures of bigger complexes, and we are actually doing this, let's say structures, for example, of alpha-actinin with in complex with larger titin complexes, or ZASP and several other binding proteins. That is on the single molecule, single particle reconstruction level. But we are also joining forces with cryo-electron tomography lab, and that is to look at the Z disks, and we are actually joining forces with two. One is the cryo-electron tomography lab, and the other one is a group which is working, which has a capacity of producing stem cell derived cardiomyocytes. And this actually allows them to control their development, their maturation. So the idea that we have is to look at these Z disks, these structures at different stages of biogenesis, and which could be easier controlled with these stem cell derived cardiomyocytes. I hope this was the answer to the question.
Yes, thank you very much. And maybe another question, whether you are planning to use any thermophilic sort of, maybe a worm or some other organism that will help you with this, just like the colleagues in Heidelberg have done with the nuclear pore using the thermophilic fungus.
I know. We would love to. The thing is that in these different organisms, then the Z disk is not the same anymore. For example, if you go from human to insects, for example, the structure of the Z disk goes from tetragonal to hexagonal. Then you have also the issue that at certain lower organisms, which still have some sort of a muscle, they don't have titin anymore, but they have chitin or other or protein. So then it would be difficult to translate back. See what?
Okay. Are there other questions from the active cameras that I see?
Yeah.
Actually, yeah. Who's
Christine? I'm regarding the fuzzy complex. I'm wondering if you can analyze the SACS data in order to characterize the structural flexibility. So I have a guess about different. Yeah. So.
We did this. We did this. I thought I showed this slide. We did the fats alone, and this we modeled as a structural ensemble, as well as with in complex with alpha-actinin, where we also used SACS in order to determine or to generate this structural ensemble.
So you get the useful information from SACS.
Yeah, I would say yes, combined with what we know from the crystal structure. So we used the information from the crystal structure where it binds. We use that in the SACS modeling. The rest we let free for EOM modeling. And we also used chemical cross-linking for as an additional anchoring points, if you want.
Thank you.
Okay. Actually, there is a question from Adriana Miele on the chat. I do wonder whether Adriana would like to ask the question herself. Adriana, are you?
Hello. Yes, I have no camera. I'm sorry. Thank you. Thank you very much. It's a really great presentation. Thank you. I had one curiosity. You show the geometry of the Z disk within the contraction and then in the relaxed state. And I was wondering whether this change was due to titin and fuzz or just drained by the actomyosin contraction and then everything else is pulled and changed.
This is several million questions.
Yes.
People have been thinking, of course, about this, and the current, let's say, hypothesis is that this is really due to the force imposed on this structure, on this complex here. And people have even been doing normal mode analysis of alpha-actinin, for example, where they showed it is actually able to do quite some gymnastics which would support this idea of changing this tetragonal to weaved basket. But I think experimental data is actually lacking on this to validate this, I need to say.
Thank you.
Oh, okay, thanks. I think for the sake of time, we have to break here. I'm extremely sorry. I had 3 or 4 questions that I will address privately because otherwise the day would get too long. Christina, thanks a lot. We enjoyed very much your presentation. And I think I should now move the control to Gabriele Tedeschi for the gruppo Proteine. Then he will introduce Nicolas Gao and his lecture. Thanks again, Christina.
Yeah, I also to thank Christina a lot because it was a wonderful presentation. And I thank also Martino for suggesting this new way to do to work together between the 2 groups. So since we thought to have one senior and one young speaker today. I asked to Riccardo Miggiano, who is a young researcher in our group, to invite the young speaker. So to invite Nicolas Gao. So I ask now Riccardo maybe to say a few words regarding Nicolas. Riccardo, can you say a few things?
Thank you, Gabriella. Can you hear me?
Yes, go ahead.
Yeah, it's my pleasure to welcome Nicolas Gao. So Nick is a PhD student in biochemistry at the City University of New York, working in the David Gerusalmi lab. So we met together, I think, Nick, two years ago when I was there in New York working on cryo-EM-based studies on proteins involved in DNA repair pathway in Mycobacterium tuberculosis. Nick's research activity mainly focused on the biochemical and structural characterization of macromolecular complexes acting at the level of DNA replication initiation. And I think that today he is going to discuss the helicase loading activity of a poorly characterized helicase loader named Dcia. It is an helicase loader of Vibrio cholerae. And I don't want to take more time, so Nick, the stage is yours. I'm going to present the PPT file that you sent me, so just tell me okay when I must change the slide. Can you see the presentation?
Yes.
Okay. Yes. So I'm going to switch on presentation mode.
Great.
Yeah.
Okay.
It's fine.
Looks good.
Yeah.
Looks good. Actually, can you go full screen? We are seeing the presenter mode where we see the notes and everything.
Okay, now is in presentation mode.
Okay, we can just move on this. So I thank you everyone for being here to allow me to share my work with you, and thank you to Riccardo and Professors Bolognesi and Tedeschi for putting this together. And so the work that I do, of course, centers around helicase loading in Vibrio cholera, and so let's get started. Okay. Okay. You can move forward.
Okay.
So shown across the top is a brief scheme of how DNA replication is initiated, and it starts in the upper left corner where an initiator protein binds to a specific sequence termed the ORIC, and this binding event causes an adjacent AT-rich region of the genome to melt, and thus a replication bubble is formed. At this point, two copies of a helicase held in complex with a loader binds to the newly formed single-stranded DNA, and then that causes the formation of a pre-replication complex that's ready to start DNA replication after a variety of biochemical events. In bacteria, these three general roles of initiator, helicase, and loader are served by the proteins named DNA A, DNA B, and the loaders have been known to be either DNA C or DNA I. Okay. Bacteriophage lambda encodes its own distinct initiator proteins, initiator and loader proteins O and P, but because it is a phage, it has to borrow an essential host factor for its replication, and in this case, it takes the helicase from E. coli. Okay. So if we focus now just on the concentration of my work, this paradigm has been used to explain helicase loading for a very long time. However, recently it's been appreciated that only 15% of bacteria have DNA C or DNA I, okay? And the other 85% of bacteria don't have these canonical loaders, okay? And so the question now becomes, for this other 85%, what is the loader? And other work done by other groups have identified a highly conserved factor in this other 85%, okay, named DCIA. Thus, the focus of my work has been to show that DCIA is the loader for the rest of bacteria. And the homolog that we have chosen to study DCIA in comes from Vibrio cholera. Okay. So the first question, is DCIA a loader? And if we look at what the properties of the known loaders, these are three properties that they have. That is, the first of which is that they have to, of course, directly bind the DNAB helicase. The second is that they have to help load DNAB onto single-stranded DNA. And third of which is that they have to help stimulate DNAB's helicase or translocation activity. And so in this initial part of the talk, I'm going to walk you through the biochemical evidence that I have that establishes these three facts. So if we first move on to direct binding to DNAB, okay. What we have on the left is a DNAB that has no affinity tag that's been passed through a nickel affinity column. And understandably, most of that protein comes out in the wash and very little of it is eluted from the column because not much of it had bound. And then if we mix untagged DNAB now with his tagged DCIA and repeat the experiment, what we do see is a drastic enhancement of the band at approximately 52 kilodaltons, indicating that DNAB has formed in complex with his tagged DCIA, and thus the complex is bound to the column, suggesting a direct interaction. So next, we purified DCIA and DNAB separately and conducted further experiments using surface plasmon resonance, okay, which we of course have Ricardo and Castro to thank. And so in this format, we have immobilized DCIA to the SPR chip and passed increasingly larger concentrations of DNAB over the immobilized DCIA, shown there in the gray bars. And if the two proteins form a complex, that should elicit a response from the SPR chip, and thus you should see the purple line go up. And indeed, that they do bind to each other, but if you also look at the places in between the gray bars, those are areas where we have stopped flowing DNAB over the chip. And what you do notice is that the purple line is pretty stable, indicating that any complex that has formed is not dissociating, suggesting a very tight interaction between the two proteins. And so if we take the association rates and dissociation rates that we see from SPR, we can calculate a KD, which indicates that they have a low nanomolar affinity for each other, very tight. So they bind each other. And okay. To move on to the next point, that DCIA helps load DNAB onto single-strand DNA, we have employed native mass spectrometry as our assay. A native mass spectrometry allows us to examine the mass of intact macromolecular complexes, and thus the spectra that native MS reads out is indicative of the molecular species in your sample. And so across the bottom, we have just DNAB mixed with this with its essential cofactors of magnesium and ATP. We see a peak at approximately 313 kilodaltons, which is, if you look up to the top, represents a hexamer, has a mass that is consistent with a hexamer of DNAB. And indeed, the active unit of this helicase is a hexamer. In the next experiment, going up one, we attempted to bind DNAB to single-stranded DNA with no DCIA around, and we see a peak appear at 346 kilodaltons, consistent with the mass of a hexamer loaded onto single-stranded DNA. However, that peak is quite weak, indicating that DNAB on its own is a poor binder of single-stranded DNA. In the topmost experiment, we have included DCIA, and indeed you see that the green peak has been greatly enhanced, indicating that DCIA has helped to bind the helicase onto single-strand DNA. And okay, to examine the stimulation of helicase activity or translocation of the helicase, we radio label a double-stranded DNA fork shown on the left. And if DNAB is able to bind to one of the free 5' arms of that fork and translocate through the adjacent duplex, what you should see is a conversion of a slow-moving band to a faster-moving band because a strand gets displaced. going from DS to SS, those two lanes. And so the way that this assay works is that we mix the fork with the proteins involved and take out some of the reaction, quench it, and run it on a gel. And from that, we can monitor the progress of the reaction by looking at how much single-stranded DNA band appears. And so, as you can see, DNAB by itself is a weak helicase. It is able to melt the fork, but not to very appreciable amounts. DCIA alone cannot melt the fork, but it is only the combination of the two to get a very strong band consistent with that of the displaced band, and thus we see here that DCIA stimulates the helicase activity. Okay, and so after we had established these three basic facts about DCIA, we conclude that it is a loader. We thought next to redo these experiments with domain subconstructs, and hopefully we can get some finer details on the system, and maybe that would lead itself to a mechanism of loading. Okay. At this point, it's worth discussing the domain architectures of the two proteins. DNAB is made up of an N-terminal regulatory domain and a C-terminal Rec-A-like ATPase domain that constitutes the motor of this protein. And in between the two domains, there's a single linking helix that is known to be important for interaction with canonical loaders. At the time of the start of this project, DCIA itself was an uncharacterized protein, and so we thought to define its domain architecture experimentally ourselves. Okay. As shown here is the predicted secondary structure of DCIA, and as we move from left to right, we can see that at the N-terminal part of the protein, there's a constellation of secondary structure elements that go from left to right: alpha, 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 beta, beta, alpha, beta. And as it turns out, this unique sequence of secondary structure elements very strongly resembles that which you would find within the NTD of the DNAA initiator. And thus others in the field have termed this to be a DNAA NTD-like domain. Following this domain, a disordered linker and two C-terminal helices are also predicted. And we thought it'd be best to see how this prediction holds up to experiment, and thus hit OK. And thus we subjected this protein to limited proteolysis using two different proteases. And indeed, the predicted N-terminal domain is protected from proteolysis, and thus we think that it is a truly globular domain that folds up. And thus that leads itself to a cartoon representation below, where DCIA looks like a globular domain with a flexible linker with two helices at its end. Okay. And so we thought next to repeat the nickel pull downs, but instead of using full-length versions of both proteins, we truncated DNAB to see if DCIA occupies a more minimal footprint on DNAB. And so if we look in the table, full length versus full length, that was the gel that I showed you at the beginning of the presentation. Those two do indeed bind, but if we try and bind full length DCIA against only the DNAB NTD, they don't bind. If we bind DCIA against, if we try to bind DCIA against the DNAB NTD and linker helix, they don't bind. But we do see that it binds to the linker helix and CTD of DNAB. Okay, continue. And thus that indicates that DCIA binds to the linker helix and CTD of DNAB. Next, we did surface plasmon resonance experiments on the right, but instead of truncating DNAB, we truncated DCIA to see if the two halves of DCIA had differential affinity for the helicase. Okay. And indeed, I showed you before that full length had low nanomolar affinity, and the two halves of DCIA understandably bind weaker, but they have asymmetric, the distribution of affinity between the two halves is asymmetric in that the DCIA CTD binds tighter than that of the DCIA NTD. Okay. Okay. And thus, unless we can keep track of that here, where we see that the DCIA-CTD binds to the helicase strongly, and the DCIA-NTD binds to the helicase weakly. Okay. Okay, so here we examine the interaction of full-length versions of both proteins by native MS. Okay, and what I'm about to show you is a time series of native MS experiments. And in the earliest time point, we see peaks on the right in orange, green, and blue that are consistent with complexes that have the weight of a hexamer or greater. And indeed, we see that one DCIA has bound to the hexamer. Okay. And as we allow the experiment to progress, we see that the larger assemblies on the right become more intense, indicating that DCIA is still binding to the hexamer, but so too do we see additional peaks on the left appear, indicating sub-hexameric assemblies also appearing. Okay. And by the end of the experiment, everything that is a hexamer or larger is almost completely disassembled, and it is the smaller assemblies that dominate. And the way we think about this is, okay, is that a single DCIA is able to bind to the hexamer initially, but there's a slow disassembly process that eventually dominates, okay, leading to disassembly of the whole hexamer. We had wondered if these smaller assemblies were competent in helicase loading, and so we next thought to try and bind it to single-stranded DNA. Okay. If you co-purify the two proteins, you can start with a disassembled preparation indicated by the molecular weights in the top spectra. You can see that there are no hexamer there. And if we attempt to add single-stranded DNA to it, we do not see the appearance of a loaded hexamer peak in green. Okay. Indicating that the smaller pieces cannot reassemble around single-stranded DNA. Okay. We next mixed full-length DNA B with the two separated halves of DCIA, and what we do see in the middle peak is a complex does form, but its molecular weight is not consistent with that of both halves of DCIA being present. Okay. In fact, it is only the CTD that seems to have bound. But the salient aspect of this spectra is that, as you see, that if we go from 5 minutes to 24 minutes, these complexes persist in that they do not disassemble. Okay. Indicating that the CTD does not compromise the stability of the hexamer ring. Okay. If we contrast this against the same experiment when full-length DCIA was invoked, we did see disassembly, and it is the difference between these two experiments that we infer or speculate, okay, that it is the DCIA-CTD that is responsible for, that is the DCIA-NTD that is responsible for the disassembly. Okay. This is data that I showed you before, indicating that full-length DCIA can help bind the hexamer to single-stranded DNA. If instead we do not use full-length DCIA, but only use the DCIA-CTD, okay, we do indeed see that a large peak still persists in that of the loaded hexamer in the top spectra in the green box, indicating that the CTD alone is sufficient to enhance binding to single-stranded DNA. Okay, and in lieu of this, we speculate that this is due to the fact that the CTD is able to modify the conformation of the hexameric ring. Maybe it opens the ring, okay? And that this is why the DCIA-CTD is able to show an enhanced loading signal in native MS. Okay. And so now we have found out that the DCIA-NTD disassembles a helicase that is unloaded from single-stranded DNA, and the DCIA-CTD is sufficient to load the helicase onto single-stranded DNA. Okay. And now if we revisit the helicase assays, I showed you before that DNA-B in the red line is a poor helicase on its own. Indeed, mixing DNA-B and full-length DCIA is able to get a strong helicase signal in the orange, but the surprising fact is that the DCIA-CTD alone is able to still stimulate helicase activity in the blue line. Okay, and we explain the enhancement going from the red to blue line by the fact that DCIA-CTD is able to load more helicase onto single-stranded DNA, and that there is a spontaneous activation of the helicase once loaded. However, the difference between the blue and orange line is explained by the absence of the DCIA-NTD, okay, which at the moment we think is able to engage the nucleoprotein complex to further activate helicase stimulation. Okay. And thus we add that activity to the NTD. Okay. And so now that we have figured out that DCIA is indeed a loader, the next biggest question is what is its structural mechanism by which it does its activities? And I thought it would be helpful to explain the established loading mechanisms within the field. And the established loading mechanisms segregate into two types. That is, one is the ring breaker type and the ring maker type. In the ring breaking mechanism, the helicase is a stable hexamer, and the binding to the loader causes two events. The helicase converts from a flat planar ring to that of a spiral, and more importantly, one of the inter-subunit helicase interfaces is opened in this event, creating a gap. And that gap then allows the admittance of single-stranded DNA into the helicase. In the ring-making mechanism, the helicase prefers to be of a subhexameric oligomeric state, but it is only through the combination of helicase and loader are the subassemblies able to reassemble around single-stranded DNA, and thus the ring is so-called made around single-stranded DNA. And of course, the question for DCIA is, what does it do? Does it do one mechanism, does it do the other, or does it do neither? And my work is currently ongoing to figure this out. Because the structural data in the field is more established for that of the ring breakers, I'll now show you the ring breaking structural data structures that are out there. Continue. And so the two ring breaking structures that we have come from the E. coli helicase bound to either DNA-C or lambda P. And so these are ring breakers. The first notable aspect of this is that the loaders assemble on the bottom of the ring, and they have a characteristic arm that reaches up the side of the helicase and latches onto the side. Okay. Hopefully both movies start. And in a moment, it's going to turn to the bottom, where you'll be able to see that lambda P does this through 5 copies of itself bound to the helicase, whereas DNA-C does this through 6 copies of itself. Continue. I've recolored the helicase here to highlight different parts of it, and I'll walk you through it. I'll walk you through this part a little slowly. The 2 shades of blue represent the 2 domains of the helicase, and of course the yellow helix in the middle is highlighted in yellow. The loader is still on the bottom here, and the globular domain is still on the bottom, but it reaches up and attaches to the side of the helicase with this characteristic red helix that you see. I want to draw your attention to the inter-subunit interface, okay? Right there. Okay. And within there's this 3 helical bundle that is important to demonstrate how the loaders do what they do. So at this interface, without the loader in gray, there are 2 helices, the linker helix from one DNA-B subunit that packs tightly against helix in the CTD of an adjacent DNA-B subunit. And when the loader comes and binds, it wrenches these 2 these two helices apart, okay, like that. And it is the repetition of this wrenching motion around the ring that causes, okay, a closed planar ring to convert to that of an open spiral. Okay. And already we can see parallels between DCIA and that of a ring breaker loader. First of which is that they occupy a similar footprint on the helicase. And furthermore, the domain architecture of the loaders of DCIA seems to be that of a globular ball with a flexible linker with a helix, which is what we also see in lambda P. Okay. So in light of all this, I synthesize all the data into a comprehensive model for how DCIA might work. So first we have the helicase by itself, okay? The binding of DCIA may open the ring or modify its architecture to a to a state that is competent for binding DNA, okay? A single-stranded DNA is now able to bind, okay? The helicase gets activated, DCI is forced to leave, okay? And then you have translocation. And this would be consistent with that of the standard ring breaking model. However, we notice other areas of departure from ring breaking. Okay. The first of which is that when the helicase is not on DNA, it seems that the DCIA is able to disassemble the helicase, and we furthermore show that the disassembled complexes cannot reform around single-stranded DNA. This disassembly is something that is not seen in any of the loaders, and at the moment we don't quite know what the purpose of this is. We speculate at the moment that this is an inhibitory state that could be used to regulate the timing of the initiation of DNA replication, but at the moment that's just conjecture. Okay. The other notable aspect is that when DNAB is on DNA, we find that DCIA is able to further enhance its translocation. And this is something that is also not seen within the existing ring breakers. And that is all I have to say. Thank you for listening.
So thank you, Nicholas, for your nice presentation. So the presentation now open for discussion. Again, you cannot raise your hand, so please turn on the camera and just ask, or maybe you can write into the chat. Any questions? I cannot see anyone. Maybe, Ricardo, you want to turn off the sharing of the screen? Yeah.
Yeah, so thank you, Nicholas, for your interesting, fantastic presentation, I could say. And I just could say that the story is becoming more intriguing if we think that mycobacterium tuberculosis DCIA protein has an opposite conformation in terms of N-terminal, C-terminal domain, because we found the two helix domain at the N-terminal position in tuberculosis. So the conformation, the globular architecture of protein are just the opposite in mycobacterial tuberculosis. So the story is becoming intriguing. I would just say, Nicholas, have you tried cryo-EM studies on the complex? Do you have structural data DCIA, DNAP, macromolecular complex? We have new insight on the transformation from structural studies.
So yeah, obviously, after doing all this biochemistry, we want to put real structures to those cartoon models that I presented. And yeah, indeed, structure determination by cryo-EM is currently ongoing. We don't have anything definitive yet. All we can just say at the moment is that it seems obviously that the complex between the two just look different in the microscope than DNAB by itself. But at this point, we don't have anything else more solid to say than that.
Okay, and concerning cryo-EM-based studies, do you think that it could be a good option to couple DCIA with a high molecular weight protein in order to increase maybe the resolution or the capability of the techniques to show the complex formation? Because I think that maybe the problem is that or you can have dissociation events in the complex formation during cryo-EM-based studies, or the problem could be that maybe the molecular weight of DCIA cannot allow you to check or to analyze the presence of the protein on the complex. Do you think it could be a good idea to couple, to make a cross-link, a coupling on DCIA protein, I don't know, maybe with an antibody, in order to increase the molecular weight of the protein in maybe helping you in solving the structure by cryo-EM? Do you think it could be a good option?
On the subject of molecular weight being too small, remember that the DNAB hexamer is about 300 kilodaltons. DNAB by itself is already well within the size regime for cryo-EM, and even if we add DCIA, that just makes it even heavier, which in theory makes it even more visible for cryo-EM. I don't think that the complex is too small for cryo-EM. As far as trying to tame dynamism in the complex, of course, we saw that full length disassembles everything, and you get a bunch of heterogeneous complexes. And so we have at the moment just avoided doing cryo-EM on the more dynamic complexes. However, native MS has shown that we can get complexes with single-strand DNA, DNAB, and DCIA that persist and are homogeneous, that persist in time and are homogeneous. And so we've been focusing our efforts on that at the moment. Yes, cross-linking would be wonderful would be wonderful to stop things from falling apart. And there are many examples in the literature, of that. But our work has not gotten to that point where we've needed to invoke that yet.
Okay, and yeah, I was referring just to increase the molecular weight to DCIA in order to have a tool to check also what is the fate of the protein along cryo-EM based studies, not just to increasing the molecular weight of the complex, because I know that the XNMR of DNAB is enough to get cryo-EM based data. Maybe having a tool in order to increase also the molecular weight of DCIA, you can have the possibility to check what is the fate of the protein cryo-EM based studies. I don't know, maybe you could also on the grid, I would say. And did you try cross-linking in cryo-EM based studies?
We have not tried. We have not tried cross-linking in cryo-EM. We have tried cross-linking on its own to see if we can stop disassembly in native MS, and indeed we can. If you cross-link full-length DCIA against the hexamer, then you do get these stable 6-1 stoichiometric complexes, and so that's always an option if we need to go there.
Okay, and just the last consideration is that it could be really interesting analyzing the protein-protein interaction interface, because if the story is the same you told about is the same you told about the phage lambda and the DNAB of E. coli, it means that we have just a small protein surface on which we can design molecule in order to stop the DNA replication in pathogenic bacteria. Because if the story will be the same of E. coli DNAB with the phage loader, it could be that we have really a small protein pocket on which we can design molecule stopping DNA replication in bacteria. Could be a tool for antibiotic design or drug design, I think.
You think you can pull up slide 22?
Sorry, Nick?
Do you think you could show slide 22?
Yeah. I'm going to present again. Oh, sorry. Okay.
There's a question from
22 is
22.
22.
Yeah.
And so obviously we don't have a structure of DCIA, but we can approximate it from that of lambda P DNA B structure on the right. And so don't advance any animations. And so the globular domain in lambda P is intimately engaged with that of the helicase. We already see that of the globular domain from DCIA not necessarily being. So you're basically only, for drug targeting purposes, you're basically only looking to target that little piece on the side up, the little arm from the loader. And so yeah, it's quite a small surface to be targeting. I have no drug design expertise myself, but I think the general opinion is that the deeper the pocket, the easier it is to target, right?
Okay.
And yes.
Okay, there is a question from the audience. Actually, Nicholas, can you read it? Otherwise, I can read it for you.
Okay.
It's a question regarding the native MS analysis. And it says, when you mentioned that these assemblies have not been observed for other helicase loaded complexes of this kind, was that done in native MS conditions equivalent to yours? Is it possible that this is assembly is due to the conditions, environment of mass spectrometry?
Indeed, we were also scared that the disassembly was an artifact of the methodology, and indeed the lack of disassembly that hasn't been seen for other helicase loaded complexes has been in standard biochemical assays, such as size exclusion and ultra sedimentation through centrifugation. So we ourselves have measured at least one of the established loaders, that is the DNA B lambda P complex, and within native MS, it too does not disassemble. And so we have at least that parallel compared to.
Okay. Other questions? Okay, I think maybe it's time, if there are not other questions, I think maybe I ask Martino. Can you hear me? Because Martino says no sound.
Yeah, we can hear you, Gabriella.
Oh, okay. Martino, what do you mean no sound? No sound with the answer, no sound with what? Martino, can you hear me? I think Martino doesn't hear, I think, anybody. If there are no other questions from the audience, I may ask, I may write to Martino, I may ask Martino to join me in say goodbye to everybody, but I don't know if Martino is
I'm trying to call him on the phone.
Yeah, thank you, because, okay, I have network problems, okay, he says. Okay, I think maybe I ask someone else from
Perché è finito il coso e la Tedeschi ti sta cercando.
Okay.
Ok.
So maybe
Benissimo. Ok, perfetto. Ciao, scusami. Ciao, ciao. Hello?
Yeah?
Yes, I just spoke with Martino and he deeply apologized, but has networking problems, as he wrote on the chat. So he just asked me to thank everybody and the speakers, the two speakers, for their enlightening lecture presentation. And I think this is a good start, and let's hope to continue in the next months.
Yeah, let's hope so. And I think maybe next time the protein group will present a senior speaker and the crystallography group a young speaker. So we share.
Yes.
And, okay. So okay, have a nice evening. Thank you to everybody, and maybe Happy New Year, let's say that, because Happy Christmas and Happy New Year, since we are very close to Christmas, actually. Okay?
Yes.
Okay, thank you again to the speakers.
Maybe next time we should take a picture, but it's difficult now. There are only few online with their face.
Yeah.
And we would like also to say that this presentation has been recorded, so everybody can, look back again, and maybe within the two groups, the Italian Crystallographic Association and the Protein group, this can also be spread to the PhD students.
Yeah, definitely. This is very good, actually, and yeah. Okay, so
Martino say, let me add here my thanks to the speakers and to Gabriella for organizing this first joint webinar.
Okay, thank you, Martino, as I said before, for suggesting this kind of joint venture between the two groups. So, as we said already, it was a very nice start. So, goodbye to everybody. Okay?
Okay, bye-bye.
Ciao.
Bye. Thank you for the invitation.
Ciao Cristina.
Ciao, grazie dell'invito, veramente.
Ottimo.
Grazie.